the major impact is it affects the animal performance. And the cattle with liver abscesses don't eat as much, don't gain as much, and their feed efficiency is reduced. And that is the really major impact on the economics of the feedlot, of the beef industry. In addition to that, of the animal performance, it also impacts on the carcass yield. And there is sometimes the liver abscesses lead to adhesions to the surrounding organs and tissues, and there is a lot of trimming that needs to be done to get rid of the abscessed liver, and that leads to reduced carcass yield, and sometimes it does affect the quality of the beef too. So economically, it affects one, there's liver condemnation. Second is animal performance is affected. And second, it impacts on the carcass yield.
Welcome to another episode of the Beef Podcast Show. My name is Dr. Stephanie Hansen. I'm a feedlot nutritionist here at Iowa State University. And our guest today is from Kansas State University. Dr. T.G. Nagaraja is a professor of microbiology at K-State. He has a Bachelor of Veterinary Science and a Master's in Veterinary Microbiology. His research interest is in the field of gut microbiology of animals, particularly the rumen of cattle. His investigations have focused on the role of microbes in ruminal function and dysfunction, particularly in animals fed high-grain diets. He uses a blend of both basic and applied studies and collaborates extensively with epidemiologists, food microbiologists, molecular biologists, production animal specialists, ruminant nutritionists, and pathologists. So welcome to the show, TG.
Thank you, Stephanie.
Well, you and I definitely kind of run in some of the same circles. So we see each other occasionally at meetings. And one of the things that we both have in common is a topic I know we're going to talk a lot about today. But before we get to that, why don't we start by having you tell our listeners a little bit about your story? How did you get to where you are today and to have this important role in the beef industry?
I'll be happy to, Stephanie. I came to this country in 1972, right after I completed my master's in veterinary microbiology. And in fact, one of the reasons I came to K-State was when I did my master's degree, I was asked to give a seminar. And I chose the topic of bloat in cattle. And in fact, the title of that seminar was Role of Rumen Microbes in Bloat. Believe it or not, that was my very first kind of a public presentation to a group of people. I was profusely sweating. I still remember that. And when I went into the literature search on that topic, I found most of the studies came from Kansas State University. And in fact, the person who did the work, who passed away a long time ago, and who was my mentor when I came to this country, is Earl Bartley. He was a dairy cattle nutritionist and has done a lot of work on on bloat. And in fact, the drug we use to control bloat called Bloat Guard, which has the ingredient poloxylene, which is a detergent, still widely used in the industry for control of legume bloat. And he was the one who discovered that compound as a treatment for bloat and as well as prevention. So that was the reason why I applied to K-State to do my PhD. And I came to this country 72. I was a graduate student in biology. That's where the microbiology department is located in this university. And my research was on rumen microbes. So I worked with both the microbiology department as well as the dairy science, which then became part of animal science. Worked with dairy science. That way I had two major professors, one a microbiologist, another a nutritionist. As you would know very well, ruminant nutrition is a combination of nutrition and microbiology. So I got best of both worlds. And so that's how I started my PhD career at K-State and finished my PhD degree in 1979. And then I was offered there was a position that was open for a rumen microbiologist in the Department of Animal Sciences, and I applied, and that's how I stayed in this country. So that's how my research career started, actually, in 70s, long time ago.
So you're so right. We always teach students that as ruminant nutritionists, we're really feeding the bugs, right? And of course, feeding the microbiome is the better way to say that, because everything that we do to the ruminant animal is first affected by whatever changes happens to that rumen microbiome. And so I think that's it. That's the crux of ruminant nutrition, right? How do we not screw up what's going on in the rumen as we feed the ruminant animal? Did you have a livestock background before you came to the U.S., or was that your first experience?
I came from a city called Bangalore in India, and I only had a cat as a pet one time. And my exposure to cattle was in the veterinary school, and food animal medicine was not that well advanced in India at the time. So I had a minimal exposure and had no concept, believe it or not, had no concept of beef cattle at all. So cattle in India was mainly or exclusively for milk production. So when I came to this country, I realized the importance of beef cattle and the importance of beef industry, particularly beef cattle industry, particularly in the state of Kansas, which is as you may know, is the number one agriculture commodity in Kansas.
Yeah, absolutely. Kansas is definitely one of our powerhouse states when it comes to beef production. I think it's so important for people to understand, because I see this all the time as an animal science professor, and I'm sure you see this all the time at K-State. We increasingly get students who come in who have less and less agricultural background and livestock experience. And it's so important to, not think anything less of those kids, right? Because they might be some of the ones that are most open-minded about the opportunities. They're most eager and ready to learn. Oftentimes they don't come with some of the bad habits that we might have learned if we did grow up with cattle, like cattle handling and other things. So they're so fun. And it's so cool to see somebody like yourself who did not grow up on a beef operation, but has now had such impact on modern beef production here in the US because of what we're going to talk about next. So let's talk about kind of, if we were going to say in one sentence what your research career was, we could probably say he was the guy that helped us learn a lot about liver abscesses.
Well, I started working on liver abscess in the mid 80s. In fact, that's sort of an interesting story, and I use this as an example to tell graduate students when I teach a graduate course. My mentor, Herb Bartley, was teaching a graduate course called Rumen Metabolism. And he passed away in 1983. And I was just hired 2 years ago as an assistant professor in rumen microbiology. And so the head of the department automatically assumed, since I worked with Bartley, that I would be the logical person to teach rumen metabolism. And when I started teaching rumen metabolism, I had to read a lot more about rumen than what I was doing research on before. And when I was reading about ruminal dysfunctions, and came across the topic of liver abscess, and again, believe it or not, that's the first time I knew that liver abscess is a problem. I did not know that before. And when I read more and more about what was the information on liver abscess, it intrigued me, because here is a situation where a bacterial species that lives in the rumen of cattle and has a beneficial role to play in fermentation, in ruminal digestion, is able to cross the rumen wall, get into the blood, and reach the liver and becomes a pathogen, causes abscesses. And that really attracted my attention. And then I started realizing that a lot more work needs to be done. And that's how I got started on it. So wrote a proposal and submitted to a company, and they gave me money to start a project. So I had a PhD student at the time. And that's how my career, research career on liver abscess started, because I was teaching a graduate course and I came across this topic. And the reason I say that that's interesting is because I tell the graduate students, if you really want to be a good researcher, you have to be a teacher at the same time. You just cannot be 100% researcher, in which case you become specialized in one small, tiny area and you don't know anything about anything else. Whereas teaching allows you to keep up with what is happening in that science in the field. And that gives you a lot more ideas to do research and find new problems to solve. So that's how I got started on my liver abscess.
So let's start by helping the audience understand a little bit more about why we care about liver abscesses, right? So, what are some of the physiological ramifications on the animal, and then maybe some of the economical ramifications to final hot carcass weight, or things that the packers may not be happy about liver abscesses, before we really get into some of the why they happen and how we might overcome them?
The importance of liver abscess is it's a problem of feedlot cattle, and because of high grain feeding, although it could happen in cattle in grazing a lush grass or a pasture, and so it does not have to be entirely a high grain diet. It does happen in other cattle too, but it's more common in cattle fed high grain diet. And so the liver is abscessed, and what is again interesting is the animals that have liver abscess do not show any clinical signs whatsoever. So the only time we detect liver abscess is when the animals get slaughtered in the slaughterhouse. And the importance of this is, obviously a liver with an abscess or even a scar tissue gets condemned. And that obviously impacts the economics of a packer in the industry. But that's really a small part of the economic implication. The major impact is it affects the animal performance. And the cattle with liver abscesses don't eat as much, don't gain as much, and their feed efficiency is reduced. And that is the really major impact on the economics of the feedlot or the beef industry. In addition to that, of the animal performance, it also impacts on the carcass yield. And there is sometimes the liver abscesses lead to adhesions to the surrounding organs and tissues, and there is a lot of trimming that needs to be done to get rid of the abscessed liver, and that leads to reduced carcass yield, and sometimes it does affect the quality of the beef too. So economically, it affects one, there's liver condemnation. Second is animal performance is affected, and second, it impacts on the carcass yield.
Well, there's kind of an interesting paradigm that we also see with liver abscesses and cattle performance, right? And that's actually that A pluses, which would be the highest, some of the big, maybe even open abscesses, things like that, those are very severe and they can definitely have detrimental 20, 30, 40 pounds reduction in hot carcass for that final finishing weight. But having some small abscesses or scars that would be suggestions of prior abscesses, smaller ones, oftentimes we actually see that in some of our best performing cattle. And so we've always kind of tried to balance this like knife's edge, right? To say, well, we want to push them hard enough that they're gaining the most optimally, most efficiently, but not so hard that we tip them over into too many abscesses because of all the negative impacts on that, on trim and the condemnation of the liver in the track.
You're absolutely right on that, Stephanie. And so in the industry, we categorize liver abscesses based on the size and number of abscesses in the liver into three categories, like A minus and A plus. A plus would be the one that what you just described, which is the liver has a large abscess or multiple small to medium-sized abscesses. And whereas the A or A minus may have one small abscess or a couple of small abscesses, medium-sized, maybe a scar tissue. The A plus liver is the one that causes significant impact on the animal performance and carcass yield, because the A plus oftentimes leads to adhesion. And one of the organs that is very close to the liver is diaphragm. And so even a small adhesion leads to condemnation of diaphragm in a slaughterhouse. And that's a major loss to in terms of carcass yield. So the animals with minor or mild abscesses, one or two, in fact, they don't have any impact on the performance at all. They do as well as cattle with no liver abscess. Sometimes, in fact, they do better than the ones with no liver abscess. And as you said, that's an indication that we have gotten the maximum out of cattle with our feeding program in terms of performance.
So let's talk a little bit about some of your discoveries that you've made over the years. So talk to us a little bit about some of your identification of who might be the bacteria that are most likely to be found in abscesses. So we think they're the most likely suspects to be our problem children here that have caused the situation. And then we can kind of go from there and talk about some of the things that you've maybe learned over the years about maybe where the damage is occurring and maybe eventually how we might be able to help control it.
Sure. In fact, liver abscesses, we describe it as a polymicrobial infection, meaning it's a mixed infection. It is not just caused by one bacterial species. But there is one that is, in our experience, is almost always we find it in the liver abscess. And that's the organism called Fusobacterium necrophorum. It's an anaerobic organism like any other rumen bacteria. They live in the rumen, but not at a very high concentration. Its concentration is about 100,000 to million cells per gram or per ml of rumen contents, whereas the dominant bacteria would be in billions typically. But this Fusobacterium, as I said before, is a beneficial organism. And what it does, there are a couple of things it does in the rumen. One is to ferment lactic acid, which is a product of starch, which is grain. So grain fermentation leads to lactic acid production. And because lactic acid is very acidic, it can bring down the pH lower than normal, in which case the rumen will go into acidosis. So the lactic acid has to be metabolized as rapidly as it is produced to prevent the drop in rumen pH. Fusobacterium necrophorum is one of the bacterial species in the rumen that does that, takes lactic acid and converts that into acetic, propionic, and butyric acids, which are weak, which are not as strong as lactic acid. They're not likely to lower the pH much. And those are the acids the animal can use for their metabolism. They get absorbed from the rumen and available for the animal tissue. So that's one of the functions of Fusobacterium, to ferment lactic acid, to prevent lactic acid accumulation. Second one is it can digest proteins into peptides and amino acids, and then can take amino acids and ferment that into produce ammonia, and again acidic, propionic, and butyric acid depending on the amino acid. So it's a protein digesting peptide and amino acid fermenting organism. In fact, among all the amino acids it ferments, the one that is the dominant one is lysine. So we think Fusobacterium necrophorum is a major lysine fermenting organism in the rumen. As a nutritionist, you would recognize that lysine is a limiting amino acid, at least for dairy cows. The deaminating lysine will reduce lysine availability to the animal. So in one way, that's a negative aspect of Fusobacterium necrophorum, that it breaks down lysine. But there are other ways of preventing that from happening. So overall, it is beneficial. So what happens so that's in my opinion, is a primary causative agent. Then we find another organism called Truparella pyogenes, which used to be called Archanobacterium pyogenes, and the bacterial species has undergone 3 or 4 name changes in the last 30, 40 years. So Truparella pyogenes, in our experience, we find that in about 15 to 20% of the liver abscesses. And in some cases, in some situations, its prevalence goes higher than 20, 30%. And Truparella pyogenes, we think, is a secondary organism. So in a way, the Truparella pyogenes and Fusobacterium necrophorum kind of have a synergistic interaction. And as to give an example of that synergy, Truparella pyogenes will produce lactic acid from glucose. And the lactic acid is a substrate for Fusobacterium necrophorum. So in a way, it feeds the Fusobacterium necrophorum. So that's why it's almost always associated with Fusobacterium necrophorum. In the number of other situations where we see those two bacteria together. So that's the second one. And then about 15 years ago, when we were studying the bacteriology of liver abscess of calf-fed Holsteins, for our surprise, for the first time we found, this was in 2015, found Salmonella organism. And till 2015, believe it or not, we have cultured thousands of liver abscesses, never found Salmonella, and for the first time we identified Salmonella. And my initial thought was maybe this is associated only with Holsteins. But later, when we looked at a study where we looked for Salmonella, then we found in other cattle also, in the crossbreds also. So in our experience since then, whenever we analyze liver abscess, we look for Fusobacterium necrophorum, we look for Truparella pyogenes, and then we also look for Salmonella now. And we find those Salmonella in about 25 to 30% of the liver abscesses. At this stage, we don't know exactly whether Salmonella is a causative agent or it just happens to find a place to live and lives in abscessed liver. And Salmonella is typically in a microbiology, we consider that as an aerobic organism. But because it lives in the gut, it can grow under anaerobic conditions as well. So I'm not surprised it is able to grow and live in a liver abscess where we think it is anaerobic condition for Fusobacterium necrophorum to be able to survive and grow. So those 3 would be the top 3 organisms. But my opinion is the real causative agent is Fusobacterium necrophorum. So if you want to control liver abscess, our target should be on that organism.
Okay, so I want to talk about methods to control Fuso in just one second. But my first thought when you talked about Fuso being a lactate utilizer was thinking about Megasphaera elsdenii, right? And so my question is Fuso pretty much always present, whereas Megasphaera takes a little bit of a lag time to come up in cattle as we transition to high starch diets? Is that part of the challenge here, or how do those two compare?
Well, actually, there are about 5 or 6 different species of rumen bacteria that can ferment lactic acid. And as you said, Megasphaera elsdenii, and there's one other organism called Selenomonas ruminantium. Those 2 are, in fact, the dominant ruminal lactate fermenting organisms. And their numbers are in billions, particularly in cattle adapted to high grain diet. Whereas Fusobacterium necrophorum, as I told you before, their numbers are not that high. It's about 100,000 to a million, at the most 10 million cells per gram. So it's a lactate fermenting organism, but its contribution to lactate fermentation is not as great as Megasphaera elsteinii and Pseudomonas ruminantia. So it is there, it does ferment, but if we get rid of it, in my opinion, it would not slow down lactate fermentation because there are still other bacteria that can metabolize lactic acid.
Okay. So correct me if I'm wrong, but I think you've done some work actually trying to create a vaccine against Fuso and tested that in cases, or some folks have done that?
Yes. In fact, when I started the research on liver abscess, at the time, there was no vaccine available at all. So my initial goal was if it is a bacterial infection, we should be able to develop a vaccine. And that's how my research on liver abscess started to look for a vaccine. But that has still not been able to achieve an effective vaccine to control liver abscess. At one time, we did have a vaccine. It was commercially available. This was in early 2000, but it could not compete with tylosin, which is the antibiotic that feedlot industry uses to control liver abscess. So it went off the market, and the efficacy of the vaccine was no better than antibiotic. In other words, it did not completely eliminate the problem. It did not completely prevent it. There were still cattle getting abscessed even when they were vaccinated. So it reduced the prevalence, but did not eliminate the problem. So we are trying to improve that vaccine. So that is still my goal, that to develop a vaccine that is 100% effective. And if we come up with that might solve liver abscess problem in the industry. But obviously, the reason why we have not been able to achieve is because it's a lot more complex than a simple one bacterium and make a vaccine with that bacterium.
Right. That is honestly, liver abscesses is such an attractive thing to study from the outside looking in. And then I think once you dig into the literature and you realize that it's such a multifocal problem or challenge, right? That it's like, is it a function of microbes living in the soil in the feedlot pen? Is it a function of the rest of the dietary components? Is it the function of the genetics of the animal that we have very little idea how host genetics are influencing some of those microbial populations? So let's talk a little bit about some of those factors. So what are some of the things that you have studied over the years, or that others who have looked at liver abscess prevalence have used for approaches to try to figure out, what is causing some pens of cattle, some sources of cattle, some feedlots to have greater prevalence of liver abscesses than others?
So when we started working on liver abscess, and with the vaccine as my goal at the time, one of the things I wanted to develop was an experimental model to induce liver abscess in cattle, because that way we can test a product or test a vaccine. Otherwise, we had to go to a feed yard to test products or vaccination, and that would be expensive. Plus, you have no control on how many cattle will get liver abscess. So one of my earliest findings was that we could use ultrasound, to detect liver abscesses in cattle as a diagnostic method. And remember, I told you that the cattle with liver abscess don't show any clinical signs. So when we walk into the feed yard, we don't know which animal has liver abscess, which one is not, till they get slaughtered. So we thought, let's see whether ultrasound could be used, and we showed that we could use it, but it is not 100%. I should say, in all cases, we can't detect liver abscess with ultrasound. If the liver abscess is present on the right side of the liver, and we can scan that part of the liver which is exposed to which is on the right side below the skin muscles, easy to scan, easy to spot. But not all liver can be scanned by ultrasound because some of the liver lobes are covered by lungs, and obviously the liver on the visceral side they will have on the left side of the liver. If it is abscessed, ultrasound will not pick it up. So what we showed was ultrasound. If you see an abscess, then you know it is abscessed. If you do not see it, does not mean the liver is not abscessed. So there are a lot of false negatives. But when we were scanning the liver, we were able to detect the portal vein very prominently. And that gave us an idea, maybe we can use this to inject Fusobacterium directly into portal blood. And because the distance from the skin to the portal vein is about maybe 4 to 5 inches at the most. So we took a 6 inches long needle, and we were able to tap into the portal vein, then insert a catheter. And you could do this all by watching the scanning the liver on an ultrasound. That way you can see the needle going in, you can see the catheter being inserted, and then we can inject the bacteria directly into the portal blood. And we were able to, and those cattle developed abscesses within 3 to 4 days after that, what we call ultrasound-guided intraportal inoculation method. So that's how we were able to use that model to test some of the vaccines we had developed at the time. That way we don't have to wait to go to a feed yard to wait for 3-4 months before we see the results. So that was a major finding, and also that gave us a lot more information about how liver abscesses are induced, and as to how a bacterium that is beneficial can go to the liver, can become a pathogen. So that was a major finding. And the other major finding would be, in order to understand how what we call pathogenesis, how a bacterium can cause infection. If you think of the liver, as you know, is a very defensed organ. There are there's a lot of blood flow, a lot of white blood cells flowing into the liver, and the liver is loaded with macrophages, the Kupffer cells. So liver is a very protected organ, which is the reason why it can detoxify a number of things. So the question I asked was, how could a bacterium survive in the liver, an anaerobe survive in the liver? Because liver is highly oxygenated. And so that led us to look for how the mechanism by which an anaerobe can survive. And so thought about what kind of virulence factors the bacteria has to produce to be able to withstand the defense mechanism of the liver. And there was a indication that it produces a protein exotoxin that kills white blood cells that are cytolytic to white blood cells as well as Kupffer cells. So the protein is called leukotoxin because it affects leukocytes. And it also is a gram-negative organism, so it does have endotoxin. And so endotoxin can contribute to inflammation of the infected tissue or organ. So the reason why an anaerobe can go to an organ that is highly aerobic and survive and grow and cause infection is because of leukotoxin. So that gave an idea, why not we use leukotoxin, which is a protein, meaning antigenic. Why not we make a vaccine with leukotoxin in it? That way the animal will produce antibodies against leukotoxin. So if the Fusobacterium goes to the liver, releases leukotoxin, antibodies can neutralize it, and then the liver can handle the bacteria. So that was the concept behind our vaccine that we developed based on what we call leukotoxoid vaccine, meaning a leukotoxin that was inactivated to lose its biological activity but retain its antigenicity. So we had to screen number of Fusobacterium necrophorum strains and identify a strain that produces a lot of leukotoxin, because the amount of leukotoxin they produce varies from strain to strain within a species. So we were able to get a patent for that strain that produces a lot of leukotoxin. Then we had to conduct studies to optimize leukotoxin production. The right medium, right culture conditions, right duration of incubation, that way there's maximal production of leukotoxin. And then we had, in order to test our theory that if we have leukotoxin as an antigen, we can prevent liver abscess, we had to develop the liver abscess model to test it. That's how we used ultrasound. And so that's how we were able to develop a vaccine that could be used to prevent liver abscess. And the fact that it is not 100% effective means there are other factors involved besides Fusobacterium necrophorum and leukotoxin alone. And that's what we are focusing on these days.
So is leukotoxin, would it only be produced locally in the liver, or is it something that you might expect to pick up like in a systemic blood sample?
I think the leukotoxin is produced locally. I'm sure it is produced in the rumen. Because it's a protein, it will get digested right away. But the effect of leukotoxin would only be would only make sense if the organism is in a tissue. So from the rumen contents, Fusobacterium necrophorum first gains entrance into the rumen tissue, rumen wall. And we think it grows in the rumen epithelium. And the reason why it's able to grow is because it can, again, prevent phagocytosis by producing leukotoxin. Remember I told you the Fusobacterium necrophorum in the rumen is very low concentration. But what happens when it goes into the rumen epithelium is it grows and the concentration goes up, increases. In a way, I call the rumenitis that results from that infection is a prerequisite for the bacteria to go into the liver to cause liver abscess. And so one of the reasons why the bacteria can go into the rumen epithelium is because the rumen epithelium is somewhat damaged. And that damage happens because of acidosis. So which is the reason why we think ruminal acidosis and subsequent rumenitis are predisposing factors to cause liver abscess. So that's why most nutritionists, feedlot nutritionists, they lump acidosis, rumenitis, liver abscess as one syndrome. They call acidosis, rumenitis, liver abscess complex, because the acidosis leads to rumenitis, rumenitis leads to the liver abscess. We don't see leukotoxin in the blood of cattle, but we see antibodies against leukotoxin in the blood. So in fact, I thought maybe we could use a leukotoxin antibody titer as an indicator of whether animal is abscessed or not, as a potential diagnostic test. We found out that there was not much correlation between antibody titer and liver abscess. And that may simply because it's in the rumen epithelium if there's rumenitis. So an animal could not have a liver abscess, but could still have rumenitis. There will be antibodies against that. And also you said a few minutes ago that it's the soil, and Fusobacterium necrophorum is also in the soil. So, but we don't know whether it's a soil bacterium or soil got contaminated from the manure, because Fusobacterium is in the rumen, it's in the feces, in the manure. Hence, soil is contaminated. which is also the reason why the same organism causes foot infection in cattle, foot rot. So it's the same causative agent that Fusobacter necrophorum causes foot rot, causes liver abscess. And there's one other infection in cattle that we see caused by the same organism. There's calf diphtheria, which is necrotic laryngitis, typically seen in 1 or 2 year old, typically 1 year old calves. And it's also part of the normal flora of the mouth of cattle. So in a way, it's present in the mouth, it's present in the GI tract, it's present in the soil. So that's why in most cases we call that liver abscess as an endogenous infection, because the organism is already in the animal. It just comes into the liver to cause abscess.
So when you had your vaccine against Fuso, did you also see reduction in foot rot?
In fact, at the time, the company had some evidence to indicate foot rot was reduced, but they did not get label claim for foot rot. So it was never meant for foot rot, because there was no study done to show the efficacy in a foot rot control. But I think any vaccine, if you were to develop an effective vaccine against liver abscess, I'm 100% confident it will reduce foot rot.
Well, it's really interesting, right? Because like where I am in the upper plains, we largely do not feed tylosin in the diet because our rations don't include steam flake corn. They're not quite as hot, right? They're not quite as calorically dense, maybe as some of the southern plains rations. But we definitely have foot rot issues, right? Especially as we think about a part of the country that's wetter. We have deep bedded pits. We have other, slatted floors where we get maybe more physical damage to the hoof. We create opportunity for that infection to occur. So that's really interesting. I had not realized the connection that Fuso was a part of the foot rot problem.
Absolutely. So.
Okay, let's talk about the elephant in the room, and that is these dairy beef creatures that are hitting the feedlot now, right? So we're just starting to learn a little bit about some of the prevalence of liver abscesses in them, and I would definitely say that we don't have enough data yet on that. But what is your thoughts on, are we likely to see a lot more liver abscesses in these calves that come from dairy cows but have been used for, beef semen? So tell us your thoughts on dairy beef.
Well, I just got exposed to that part of the industry 2 years ago, the beef on dairy. As you said, there's not much data available. In fact, all the report about higher liver abscess incidence in beef on dairy. The estimate is about 2 to 3 times more than the crossbred beef cattle, which means about 60 to 70% of those cattle will have liver abscess. All those reports are anecdotal, and there's no published data, and that's why we have started working on those animals, those cattle. And I think one of the reasons, in a way, beef on dairy is similar to calf-fed Holsteins, in the sense that you take the male Holstein calves, and right after weaning, they are exposed to high-grain diet with very little roughage, and they're on a high-grain diet for almost 300 to 350 days. Compared to an average 150 days, 160 days for crossbred beef cattle. So same thing happens to beef on dairy. So they're given a diet that is highly fermentable right after weaning, not much roughage. And from the calf ranch, they go to the grower yard, where they may get little more roughage, and then they end up in the feedlot. So by the time they're born, till they reach feedlot slaughter, about 300 to 350 days, and most of those time they are on a high grain or grain diet with less roughage or little roughage. So I think the major reason why we see high liver abscess is because days on feeding, a lot longer than most beef cattle. So as a result, there is more chance for acidosis, more chance for ruminitis, and that's the reason why we see more liver abscesses. And that's what we think it is. We don't know exactly whether there are other factors besides just on days and feeding is responsible for higher incidence. We don't know that. And one of the things we are doing right now is to look at the bacteriology of liver abscess of beef on dairy to see is there anything different in those cattle compared to crossbred beef cattle. So at least we can rule out it is not because of there are different bacterial species involved in causing liver abscess.
Do you know if on dairy cows, if we think about like whenever a mature Holstein cow gets culled from the dairy operation, do they have liver abscesses? Is it liver abscesses more prevalent in dairy cows, period?
In fact, there are a couple of studies done to look at liver abscess prevalence. In fact, I was involved in one of those about 20 years ago. The culled dairy cows that are slaughtered for beef, they do have liver abscess. In fact, the prevalence is about the same as in feedlot cattle. 25 to 30% of them have the liver abscess. Remember, these cattle don't get tylosin. Where we can feed tylosin to feedlot cattle. And I think, because dairy, they go through that change in feeding. When they're, lactating, they get a high grain, although there's enough roughage, but their feed intake is much higher than most cattle. And then when they're pregnant and not lactating, they're typically given a high forage diet. Then when right after parturition, and they get more grain. So they go through a lot of those changes, major dramatic changes in the diet in their life cycle. And I think those may be the reasons why they do develop liver abscesses.
Well, and I think if we tie it back to our earlier conversation where we, kind of think about the beef cattle that had some abscesses, but not A+ abscesses, do have better overall performance. Is that kind of potentially the same thing happening on the dairy cow? Because, the dairy cow is basically a metabolic racehorse, right? Like she's been, and like you said, she has huge intake. She has huge metabolic demands on her. She's outputting all that milk. That'd be really interesting to see if we could tie back that prevalence to, milk yield or milk efficiency or whatever the dairy people would use for a metric.
Right. That's an interesting observation because I don't think anybody's looked at it, whether any relationship between cattle that had one or two abscesses after they're culled, to go back and look at their lactation cycle and milk production data. So it is possible. Again, if you see one or two mild abscesses, again, indicates the animal was performing at its peak, at the best it could do.
Right. So as we approach the end of our time together here, I want to give you the opportunity to answer this question, and that is, what are you most excited about right now in the field of liver abscesses? What do you think is the thing that might be just around the corner for discovery?
Well, obviously vaccine is still the ultimate goal, because if you have an effective vaccine and it's easy to administer, then it would replace use of an antibiotic in the industry, because tylosin, as you know, is what we consider as a medically important antibiotic. Because although tylosin is not used in humans, an antibiotic very similar to tylosin is erythromycin, widely used in humans. So obviously the vaccine is still my dream intervention strategy, if we can accomplish that. But one of the things we are working on right now is to see, we talk about Fusobacterium necrophorum, rumen, acidosis, ruminitis. The question that have been asked often is the hindgut of cattle. And I know a lot of nutritionists would agree when you feed a high grain diet, and particularly, as you said, in a steam flake corn or any fermentable grain, the acidosis could also extend to the hindgut, because hindgut is exactly like rumen, except it is less buffered than rumen is. And so we could potentially be creating hindgut acidosis. And in fact, some nutritionists believe that may be the real problem, not the ruminal acidosis. And we have found that Fusobacterium necrophorum also lives in the colon of cattle. So the question is, could hindgut also be the source of bacteria that come to the liver to cause liver abscess? And we don't know the answer to that yet. We are working on it. And some of the data that we have generated so far indicates that hindgut does seem to have a role. And the reason why it's important is because if we know hindgut could also be the source, so we have one more target to look for intervention to prevent Fusobacterium from crossing the hindgut epithelium to come to the portal, because all the blood from the GI tract, as you know, has to go through the liver. So it does not matter whether the Fusobacterium is in the rumen or in the hindgut, they end up in the liver to cause liver abscess. So that's in a way, a study that we are doing to answer a question that has been frequently asked: does hindgut have any role to play in liver abscess? The second interesting aspect of our finding of our study is the Fusobacterium necrophorum in a species, but there are two different types of Fusobacterium necrophorum. And one type, and these are called biotypes, used to be called biotypes. Now we have given a subspecies name. Those names are not that important. Anyway, of the two types, there's only one that is frequently found in liver abscess, not the other one. And we call it subspecies necrophorum or biotype A. That's the one that produces more leukotoxin than the other type. And that's the reason why it causes liver abscess. Whereas the subspecies funduliform or biotype B does not produce leukotoxin, so it does not cause liver abscess. So that lives in the rumen. And we were using culture method to enumerate those population of those bacteria or to identify them. So recently we have developed a PCR assay that can distinguish between those two subspecies or biotypes. What we found, to our surprise, was not all cattle rumen contains the type of Fuso that causes liver abscess. So only about, at the most, 50% of the cattle contain that organism. Other 50% don't. So when we say we should target Fusobacterium necrophorum to prevent liver abscess, now I would say we should target that type of Fusobacterium that causes liver abscess, not the other type. The other type is beneficial. So the subspecies necrophorum or biotype A is the real pathogen, real culprit in causing liver abscess. So our focus in preventing liver abscess should target the subspecies necrophorum, the one that causes liver abscess. Now, what we don't know is why only some cattle have, others don't. And I still don't know whether the cattle that have this are the ones that develop liver abscess and the ones that don't develop liver abscess. We don't know that. Because those are the studies we need to do in order to come up with a little more understanding of liver abscess. Again, the bottom line, Stephanie, is, and I tell this to many, I've been working on this problem for the past 30, 35 years. We are still trying to finding out new things that we did not know before. In a way, that's what research is, right? So when you ask a question, you design a study to find an answer. A lot of times you find an answer, but you also raise 2 or 3 more questions. And that's how the research keeps continuing. So 35 years of liver abscess, there are still unanswered questions about liver abscess.
Absolutely. Well, I think that was a really great recap. And I guess I would just, say for our listeners, just practical ways to prevent liver abscesses, going back to what TG has talked about, tylosin, if that's an availability, if you're more in the steam flake diets, managing roughage levels, and then bunk management, anything that's going to prevent these bouts of acidosis that might cause gut damage. And just to help listeners understand too, I'm totally in the camp that thinks that hindgut damage might be more impactful than rumen, because rumen is basically as tough as shoe leather. It's got multiple layers of cells, a dozen plus layers of cells, whereas we've got single cell layer of protection by the time we get to the hindgut, places like the colon or pieces of the small intestine and stuff, right? So I totally believe that it seems so much more susceptible. And some of the work we've done looking at like zinc supplementation to overcome gut barrier issues potentially has some positive impacts there on liver abscesses. So it's so cool to hear from you, the story of Fuso and kind of how that has evolved across your career. Okay, so we've reached that point of the episode where we've got our 3 famous questions. So these are the questions we ask of all of our guests. So our first question is, what is your favorite beef resource?
In terms of beef research?
It could be anything. We've heard the NRC, we've heard recipes, we've heard we've had some like station reports or beef department reports. So where's your favorite place to go to find information about beef?
I do read the beef, the industry magazines on beef, the feedlot industry. Because I'm a researcher, obviously I track research publications that deal with beef cattle, and anything to do with the GI tract would interest me to read it. I think this is another lesson I try to teach my graduate students, that if you want to be a good researcher, you have to read and learn about what others are doing in your area. The more you read about, the more ideas you get about how to do or what to do in terms of research.
Perfectly. You've teed us up excellently for our second question is, what is something not related to beef that you're reading right now?
Unfortunately, I'm not reading any book now, but I tell you, my most favorite reading every day is New York Times. And because it's available online, I can get that on my iPad. I spend about half an hour every day to read the New York Times. And New York Times covers a lot of topics of interest. For example, just a few weeks ago, I read about ChatGPT for the first time. I understood what the AI and its impact on education, as an example. Used to be a voracious reader of books one time. Particularly when I'm traveling, I like to carry a book and read. But these days I carry my laptop and work on it, which is unfortunate.
As the graduate students say, the graduate students start their presentations 2 weeks before the meeting. The professor, we're typing the presentation on the plane to the meeting, right?
Yeah, that's how it works most of the time. So.
Okay, third and final question for you here is, what is a trait of someone you know that has helped them be successful?
I thought about that question. My mentor I told you about in the beginning, Earl Bartley. He was a dairy cattle nutritionist and a top-notch scientist, very well established name at the time. And I watched him as my mentor, how he interacted with graduate students, watched how he interacted with the colleagues. And the trait I learned from him is how to be collaborative, how to bring people with the different expertise into one room and discuss the project that you're working on to get input and get ideas. And that importance of collaboration in research, I learned from him. And he was very good in interacting with veterinarians, with grain scientists, and with statisticians. And so a lot of my mentoring style, my enthusiasm in research, I learned from him, our Bartley. So I give him a lot of credit. And whenever I teach human metabolism, I spend about 10 minutes to talk about my mentor, because that impression that he created at the time, and my dream job at the time was to be like him. I should be a professor, have my own lab, my own lab technicians, my own graduate students, have projects to work on. I think to some extent I've accomplished that. And he was the person who helped me develop into a scientist.
Well, I think that is an excellent way to end our time together, TG. It's been so great talking with you today, and we've really appreciated your time.
Oh, my pleasure, Stephanie. I really enjoyed it, and thank you for inviting me to be part of this.